Welcome to the Deep Dive, where we take complex topics, stack up the sources you've provided, and unearth those crucial nuggets of knowledge. We do it all while making sure you're well informed without feeling overwhelmed. Today, we're embarking on a mission to truly master a vital, life-saving field that impacts millions: transfusion therapy and biology. Our goal is to dive so deeply into this material that by the end of this deep dive, you'll feel like an elite professional in this field.
Indeed. And our primary source material, Chapter 113 from a comprehensive medical text, offers an incredibly detailed and authoritative look. It covers the science, the practice, and the intricate biology behind blood transfusions.
Yeah.
It's really a foundational text, providing the bedrock for anyone seeking to understand this field at a professional level, and we're going to unpack every concept.
That's right. We're talking about everything, from the meticulous process of how blood components are prepared to the truly fascinating, sometimes surprising world of blood groups. And crucially, the critical importance of safety and managing potential reactions when things don't go perfectly.
Our mission for you, our curious listener, is to extract the most important insights from this chapter, exploring not just the what, but deeply understanding the why behind every what. We'll connect the dots to give you a comprehensive, authoritative, and frankly, truly expert understanding of transfusion medicine. This isn't just about memorizing facts, you see. It's about grasping the profound interconnectedness of biological processes and the critical clinical decisions that save lives.
Okay, let's unpack this.
The foundation of transfusion medicine blood components and their journey.
So to kick us off, what exactly is transfusion in this highly specialized medical context? It sounds straightforward, maybe, but it's clearly far more complex than just, giving blood.
Right. At its core, transfusion encompasses the strategic and precise use of blood components, BCs. We're talking about incredibly specific preparations derived from donated blood.
Right.
While the primary goals that come to mind for many are, treating conditions like anemia, hemorrhage, or various bleeding disorders, what's truly fascinating is the broader scope. Our source highlights that occasionally these components are strategically employed to combat infections or even manage relapses of malignant blood diseases, particularly after complex procedures like allogeneic hematopoietic transplantation.
Wow.
This truly speaks to their versatility and, the depth of their application in medicine.
That's a much broader scope than many might realize. Certainly beyond just replacing lost blood volume. What exactly are these fundamental blood components we're talking about then?
So the main blood components used directly for transfusion are red blood cell concentrates, RBCCs, which are designed to carry oxygen.
Right, oxygen.
Then you have platelet concentrates, PCs, which are vital for clotting. And finally, plasma for transfusion use, which contains clotting factors and other essential proteins.
Okay.
It's crucial to understand the distinction here, though. We're talking about plasma directly given to a patient, as opposed to plasma collected for fractionation.
Fractionate.
Yeah, that's a process where it's separated into specific medicinal products, things like albumin or immunoglobulins, usually for industrial scale manufacturing. Those undergo entirely different processing steps.
I see. And beyond simply getting the right blood to the right patient, what are the overarching critical aspects that elevate this field into a specialized medical discipline? And what are the sort of guiding principles?
Well, beyond the obvious matching, three critical aspects really define the specialization and rigor of transfusion medicine. First, ensuring transfusion safety. This is absolutely paramount, a constant vigilance to protect the recipient. Second, meticulously assessing blood component quality to ensure that every single unit performs effectively and consistently as expected.
Quality control, right.
Exactly. And third, promoting evidence-based transfusion practices. This means every decision, every threshold, every procedure is rooted in the very best available clinical research and data. These three pillars demand constant attention, making transfusion medicine a dynamic field. It's always balancing profound benefit with, inherent risks.
That sounds like a perpetual balancing act, requiring incredible precision. 1.2, the journey of blood components: collection, processing, and quality. Before we get into the nitty-gritty of what's actually in the blood, let's consider the journey itself. How did these components even get prepared for transfusion? Our source provides a comprehensive Table 113-1, which effectively lays out this entire intricate process. It seems like quite the logistical feat.
Oh, absolutely. This table is central to understanding the entire logistics of blood product manufacturing. Most common blood components are collected in one of two ways.
Okay.
Either as whole blood from a standard venipuncture donation, the typical blood drive scenario, or directly as specific components via apheresis.
Apheresis.
Yeah, where a machine separates the desired component, like platelets or plasma, while returning the rest of the blood to the donor.
Right.
The vast majority of these components are homologous, meaning they come from a separate, unrelated donor, while autologous blood components collected from the patient themselves, maybe before surgery.
I've heard of that.
Yes, they were once quite common, but they've become increasingly rare now. This is largely due to, well, limited evidence-based advantages over homologous species in most situations, though they do retain significant, sometimes life-saving value for patients with extremely rare blood group phenotypes where finding a matching unrelated donor is nearly impossible.
So, if I understand correctly, whole blood, once collected, goes through a very specific and detailed process to separate it into its different parts. It's not just, split down the middle, is it?
Precisely. For whole blood, once collected, there are two main separation pathways to yield individual components. Each involves carefully controlled centrifugation steps.
Centrifugation, spinning it fast.
Exactly. One pathway involves a slow centrifugation first. This separates the whole blood into two primary layers. A layer of red blood cell concentrates, RBCCs at the bottom,
Okay.
and a top layer of platelet-rich plasma, or PRP. This PRP then undergoes a second, higher-speed centrifugation to further separate out the platelets themselves, leaving behind the plasma.
Got it. And the other way.
The alternative pathway uses a high-speed centrifugation step initially. This separates the whole blood into three main layers: packed red blood cells, PRBCs at the bottom, plasma at the top, and an intermediate layer called a buffy coat.
Buffy coat.
Yes, it's a concentrated layer containing leukocytes, the white blood cells, and platelets. This buffy coat is then processed further. It can either be pooled with buffy coats from other donations to create larger pooled platelet unit,
Okay.
or it can undergo additional high-speed centrifugation to produce specialized granulocyte units. It truly is an intricate multi-step manufacturing process, each step precisely controlled to maximize the quality and yield.
Fascinating how it all gets broken down, almost like a refinery for blood. And what about quality assurance for all these components? With something so vital, I imagine it's not just strict, but meticulously enforced at every single stage.
Absolutely. The rigor is immense. All blood components must meet stringent quality and performance standards and guidelines set by national and international health authorities. That ensures consistency and safety across the board.
Makes sense.
This involves not just well-defined processing steps, but also rigorous quality controls at various critical points throughout the entire process. There's a strong emphasis on traceability of all manufacturing steps.
Traceability, like tracking it.
Exactly.
Right.
From the moment of donor collection right through to the final transfusion into the patient. Furthermore, hemovigilance-based reporting of adverse events and incidents covering everything from blood collection to component processing and the actual transfusion is not just recommended, it's vital.
Hemovigilance.
It ensures continuous learning, identification of new risks, and ongoing improvement across the entire blood supply chain. This raises an important question. Once we've processed the blood, what specific additional steps do we take to ensure these components are as safe as humanly possible for the recipient, especially when we consider potential risks beyond just matching blood types?
That leads us directly to some key additional processing steps, doesn't it? The source highlights leukocyte reduction as a major, almost universal one now.
Yes, indeed, with the obvious exception of granulocyte concentrates and mononuclear cells, which are specifically collected for their cellular content.
Right.
The vast majority of blood components today are leukocyte reduced. This involves removing most of the white blood cells. Universal pre-storage leukocyte reduction is now widely recommended, and in many places it's even mandatory.
Yeah.
These processed components are designed to contain a minimal number of donor leukocytes, typically less than 1 to 5 million per unit, a drastically reduced amount.
What's the profound rationale for reducing these white blood cells? What specific problems, even subtle ones, does that prevent for the recipient?
Leukocyte reduction is a multifaceted and crucial safety measure with significant clinical impact. First, it significantly reduces the incidence of febrile non-hemolytic transfusion reactions, FNHTRs.
Those fever reactions.
Exactly. The common and uncomfortable fever and chills. Second, it profoundly lowers the risk of infections with intracellular pathogens like cytomegalovirus, CMV.
Well,
Yes, which can be particularly dangerous for immunosuppressed patients, as CMV often resides within those leukocytes. Third, it plays a crucial role in preventing alloimmunization.
developing antibodies.
Right, when the recipient develops antibodies against foreign donor antigens, especially HLA antigens found on leukocytes. And finally, it reduces unwanted immunomodulation, those subtle but potentially significant changes in the recipient's immune response due to the transfusion. So it's not just one benefit, but really a cascade of safety improvements.
What other optional or sometimes mandatory steps are taken beyond just removing white blood cells? Are there ways to deactivate other threats or tailor components even further?
Absolutely. We also commonly see irradiation of blood components. This is typically done using X-rays or gamma radiation, applying a precisely controlled dose of approximately 25 to 35 gray.
Gray, the unit of radiation dose.
Correct. For red blood cell concentrates, it's most often performed on units no older than 28 days after collection, and for platelets, generally on bags no older than 3 days.
What's the specific critical purpose of irradiation?
Yeah.
What life-threatening complication does it directly prevent?
Its primary purpose is to prevent transfusion-associated graft versus host disease, GVHD.
GVHD. That sounds serious.
It is an extremely severe and often fatal complication that can occur in immunosuppressed patients. It happens when donor T lymphocytes, if present and viable in the transfused product, engraft and then mount a devastating immune response against the recipient's own tissues.
Wow.
Irradiation effectively inactivates these donor lymphocytes, preventing them from proliferating and causing this catastrophic harm.
Okay, so that's critical for certain patients. What else?
Then there's pathogen reduction, an innovative process that uses methods like nucleic acid cross-linkers or specific UV illumination.
Pathogen reduction to kill germs.
Essentially, yes. This process aims to further reduce the risk of transfusion-transmitted infections by inactivating a wide range of viruses, bacteria, and parasites that might be present in the blood component. It provides an extra layer of safety, a kind of chemical or physical fail-safe on top of the stringent donor screening and testing.
And beyond these broad safety measures, what else can be done to tailor components for specific, often vulnerable patients? I'm thinking of delicate cases like pediatric patients or those with particular known sensitivities.
Good question. For pediatric preparation, blood components are meticulously adjusted and divided into smaller aliquots for low-weight recipients. This ensures they receive appropriate volumes and cell counts without being overloaded. Precise dosing is vital for their smaller circulatory systems.
Makes sense for kids.
Then plasma reduction is another important step. It's performed to prevent allergic reactions in patients who have a history of prior severe reactions. This effectively reduces the amount of residual plasma, reducing potential allergens, though it does lead to about a 10% reduction in the red blood cell content.
And you mentioned freezing.
Yes, a truly vital technique is cryopreservation. This involves deep freezing, typically using glycerol for red blood cell concentrates or dimethyl sulfoxide, DMSO, for platelets.
Why freeze them?
It's primarily used to ensure the long-term availability of rare blood groups for immunized public negative recipients or those with complex alloy immunizations where finding a fresh, compatible unit might be impossible.
For the really hard-to-match cases.
Exactly. It also ensures a continuous supply in remote locations or for specific emergency preparedness. The shelf life after thawing varies, maybe 24 hours, maybe up to 7 days for red cells, depending on the solution. But cryopreserved red cells stored properly, say in liquid nitrogen, can last for 30 years or even indefinitely. Truly remarkable.
Here's where it gets really interesting for me. What happens to these components during that long storage? Do they just sit there inertly, or can they actually degrade or change in ways that affect their quality, efficacy, or even safety when transfused?
That's a critical concept. It brings us to what are known as storage lesions. Blood component constituents are definitely not inert. They undergo a complex series of biochemical and morphological changes during processing and storage.
Okay.
This degradation is influenced by various factors. The physical stress of centrifugation and filtration, continuous contact with plastics from tubing and bags, the chemical environment created by anticoagulants and additive solutions.
Right.
They also experience gas exchanges different from aerobic breathing in the body, and crucially, they are maintained at non-physiologic temperatures, either 22 degrees C for platelets or 4 degrees C for red cells. Any of these elements can contribute to these storage lesions.
So they degrade over time.
They change. Some of these changes, these lesions, have proven to be reversible in the recipient after transfusion, meaning the cells recover some function. But others may be irreversible. Their precise clinical impact is still under extensive investigation. While they may contribute to adverse reactions, there's currently no universal consensus on the full extent of the issue.
So even the small amount of residual plasma in some blood components can carry significant risks, like potentially dangerous donor antibodies. It's not just about the cells themselves matching.
Absolutely, that's a crucial detail often overlooked. The plasma present in blood components contains donor antibodies, ABs. If these antibodies are directed towards antigens on the recipient's cells, like their blood group or tissue HLA antigens, they can indeed cause adverse events, including severe hemolytic reactions.
So plasma matters too.
Definitely. Red blood cell concentrates, due to processing, bring only minimal donor plasma, maybe 10-30 milliliter. But platelet concentrates, even with additive solutions replacing much of the plasma, still leave the equivalent of about 1 plasma unit, roughly 200 milliliters per transfused PC.
That's a lot more.
It is. This substantial volume of plasma in platelet concentrates means the presence of high-titer donor antibodies against the recipient cells is a very real and clinically significant concern for safety, demanding careful matching.
The intricate world of blood group antigens and antibodies.
This sounds like the core, maybe the most complex challenge of transfusion medicine, ensuring a perfect or at least a safe match between donor and recipient. Let's delve into what happens at the cellular level when blood components don't perfectly match, and how the body's immune system gets involved in this intricate dance.
You've hit on one of the most fundamental and complex challenges. Red blood cells, and also other blood cells like platelets and neutrophils, all express allogeneic determinants on their surfaces. These are molecules that vary significantly from one individual to another.
Like unique flags on the cells.
Sort of, yes. Transfusion, therefore, inherently carries the risk of alloimmunisation. That's the process where the recipient's immune system produces antibodies, alloAbs, specifically against these foreign determinants, essentially recognizing them as non-self. It's the body's natural defense mechanism, but in this context it can become quite problematic.
What kinds of antibodies are we talking about here? Are there different types specific to different cell types that our body can generate?
Yes, precisely. These alloantibodies are diverse and target different cell types. They include anti-red blood cell, RBC, antibodies, which are the most commonly recognized and cause hemolytic reactions.
Okay, the ones that break down red cells.
Right, but they also encompass anti-HLA antibodies directed against human leukocyte antigens found on various cells, including white blood cells and platelets. Then there are anti-human platelet Ag, HPA antibodies specific to antigens on platelets, and anti-human neutrophil Ag, HNA antibodies, targeting antigens found on neutrophils. Each type has its own distinct clinical consequences.
And what are the specific clinical consequences, the real-world impact of these different types of antibodies once they're formed in a patient? How do they manifest?
The consequences vary dramatically. Anti-RBC immunization can lead to hemolysis, the destruction of red blood cells. This can be immediate and severe, potentially life-threatening.
Right.
Anti-HLA or anti-HPA antibodies can cause other complications like fever and, notably, platelet transfusion refractoriness.
Refractoriness, meaning the transfusion doesn't work.
Exactly. The patient's platelet count doesn't rise as expected because the transfused platelets are rapidly destroyed by these antibodies. Moreover, in a critical reversal, anti-HLA and anti-HNA immunization of the donor, if their plasma is transfused, can cause the severe lung disorder TRALI in the recipient.
Donor antibodies causing problems in the recipient.
Correct. And finally, some antibodies, particularly IgG or IgM, can activate the complement system, leading to cell lysis. Certain IgG antibodies are particularly concerning because they can cross the placental barrier, leading to hemolytic disease of the fetus and newborn, HDFN.
HDFN, where the mother's antibodies attack the baby's red cells.
Precisely, often requiring complex treatments like intrauterine transfusions. 2.2 erythrocyte blood groups, the diversity on our cells.
This is where we get into the famous A, B, O, and Rh factors, the blood groups everyone has heard about. What exactly defines an erythrocyte blood group antigen, biologically speaking, and why is there such incredible diversity among them?
Erythrocyte blood groups refer to specific antigenic molecules expressed on the surface of red blood cells, and often other cells too. These molecules are genetically transmitted, inherited from our parents, and are precisely recognized by specific antibodies. The remarkable polymorphism.
Polymorphism, meaning lots of variation.
Exactly. The vast diversity and variation in these molecules from person to person is what fundamentally explains their significant immunizing potential. This potential becomes critically important in contexts like transfusion, pregnancy, and transplantation, where exposure to foreign antigens can trigger an immune response. Each individual possesses a unique mosaic of these antigens.
I've heard sort of anecdotally that blood groups can interact with the environment too, perhaps influencing susceptibility to diseases. Is that actually true, or is that more like, folklore?
No, it's absolutely true, and a really fascinating area of research. Blood groups can indeed interact with the environment and even infectious pathogens, influencing individual susceptibilities in surprising and clinically relevant ways.
Like what?
For instance, malaria is empirically observed to be less severe in individuals with type O blood compared to non-O patients. This suggests a protective effect that likely evolved in malaria-endemic regions.
Interesting.
Conversely, and perhaps unexpectedly, Group O is associated with increased susceptibility to Helicobacter pylori, the bacterium that can cause stomach ulcers.
Huh. So it cuts both ways.
It does. This highlights that blood groups aren't just inert markers for transfusion. They play a deeper, active role in our broader human biology and our interactions with the microbial world around us.
That's quite a lot of variation if they're influencing disease susceptibility. So, how many different blood group antigens have scientists actually identified in total, and how are they organized or classified?
Currently, approximately 380 different blood group antigens have been meticulously described and cataloged within the human population.
380.
It's a huge number. These are further classified within about 43 distinct blood group systems. These antigens fall into two broad biochemical categories based on their molecular structure: carbohydrate blood groups, like the familiar ABO system, and protein blood groups, like Rh and Kel. Each category has distinct structures, functions, and immunogenic properties.
And what about autoantibodies? Can your own body somehow develop antibodies against its own blood group antigens, causing problems for yourself?
Yes, unfortunately that can happen. RBC antigens can indeed become the target of autoantibodies, autoabs. These are antibodies produced by an individual's own immune system that mistakenly attack the body's own cells.
Autoimmune reaction.
Exactly. When these autoantibodies target red blood cells, it leads to a condition called autoimmune hemolytic anemia. These autoabs can be categorized based on their optimal activity temperature. Some are warm autoabs, mostly IgG, active at body temperature 37 degrees C, often directed against Rh antigens. Others are cold autoabs, mostly IgM, active at colder temperatures like 4 degrees C, typically directed against antigens in the ABO, II, or P systems. These conditions can cause significant anemia and require careful diagnosis and management. 2.3. Diving deep into carbohydrate blood groups, the ABO system and beyond.
Let's start with the most famous, the one everyone learns about in school, the ABO system. How does it work biochemically, beyond just knowing your A, B, or O? What's happening at the molecular level to create those types?
Right, the ABO system is spearheaded by two main antigens, A and B. These are encoded by two active alleles, A and B, and there's also an inactive O allele. Depending on the genotype, the combination of alleles you inherit, individuals produce one of four distinct phenotypes. AB, and O is laid out in Table 113-2. What's truly unique and fascinating about carbohydrate systems, including ABO, is their biochemical nature. These antigens are considered secondary products of genes.
Secondary products meaning?
Meaning the gene doesn't directly code for the A or B antigen protein itself. Instead, for example, the A allele encodes an A enzyme. This enzyme then acts like a molecular sculptor, binding a specific A-type sugar, N-acetylgalactosamine, GalNAc, onto a precursor molecule called the H substrate.
An enzyme builds the antigen.
Precisely. The H substrate itself is expressed by another enzyme, the H enzyme, so it's a multi-step enzymatic process that builds the final antigen, not a direct protein blueprint like with some other systems.
And where are these carbohydrate antigens found beyond just red blood cells? I assume their widespread presence affects their role in the body and how our immune system interacts with them.
You're absolutely right. Carbohydrate antigens, especially the ABO antigens, are ubiquitously distributed throughout the body, not just on red blood cells. Notably, the ABO antigens are expressed on endothelial cells.
The lining of blood vessels.
Exactly. The cells lining our blood vessels, making them genuine tissue groups. This widespread expression means they can be profoundly involved in processes like graft rejection and organ transplantation.
Yeah.
ABO compatibility between donor and recipient is absolutely critical there.
Okay.
Furthermore, these antigens aren't even exclusive to humans. They are shared by many other species, including various viruses and bacteria that we encounter daily in our environment. This widespread environmental presence has profound implications for our immune systems.
That widespread presence in the environment, particularly on bacteria, must have a significant impact on our natural immunity, right? It's not just about what blood you receive, it's about what your body encounters day to day, maybe from gut bacteria.
It does. And this is a key insight into the ABO system's unique biology. The presence of A and B antigens in our environment, especially on the bacteria of our microbiota, the microorganisms living in and on us, explains the natural synthesis of natural or regular antibodies in humans. These antibodies form even in the complete absence of any prior transfusion or pregnancy.
So you just make them naturally?
Yes. For instance, a person with type O blood naturally develops potent anti-A and anti-B antibodies without ever having been exposed to A or B red blood cells through transfusion or pregnancy. These natural antibodies possess a major hemolytic capacity because they bind and activate the complement cascade very efficiently, forming that membrane attack complex that can directly lyse red blood cells.
Which makes mismatch dangerous.
Extremely dangerous. This powerful naturally occurring immunity necessitates extremely stringent donor-recipient compatibility rules for red blood cell concentrates and whole blood transfusions, and also to a lesser extent for plasma and platelet concentrates, to prevent severe, immediate, potentially fatal hemolytic reactions. 2.4, the protein blood groups unpacking Rh, Kel, Duffy, Kid, and MNS.
Okay, shifting gears now. Unlike the carbohydrate groups like ABO with their natural antibodies, the protein blood groups are described as immune and irregular. What does that fundamentally signify about their origin and behavior compared to ABO?
It signifies a crucial difference in how our immune system interacts with them. These protein antigens are typically specific to humans. This means that immunization, the development of antibodies against them, only occurs upon allogeneic stimulation.
Exposure to foreign human cells.
Exactly. Exposure to foreign human antigens. This almost always happens through transfusion or pregnancy, or sometimes transplantation. The resulting antibodies are called immune because they develop specifically after this immune exposure, and irregular because their appearance after immunization is inconsistent.
Irregular, meaning unpredictable.
Pretty much. Not everyone exposed will develop them, or their levels might fluctuate, sometimes becoming undetectable later on. These irregular antibodies, directed against RBC antigens other than ABO, must be detected through meticulous screening before any red blood cell concentrate transfusion or organ transplantation, and critically during pregnancy, because of their potential to cause severe adverse reactions for the recipient or the fetus.
Table 113.3 seems to be central for understanding these different protein systems. Let's start with the Rh system, probably the most famous after ABO, known for Rh positive and Rh negative status.
Right. The Rh system, formerly known as rhesus, is incredibly complex at the genetic and antigenic level. It comprises nearly 56 distinct antigens.
56?
Yes, but the most clinically significant and immunogenic of these, the one we hear about most often, is the Rhd antigen, Rh1. It's the primary determinant of whether someone is considered Rh positive or Rh negative.
How are the Rh antigens encoded genetically? What are the key genes involved in this complex system that lead to being positive or negative?
The Rh antigens are coded by two closely linked genes located on chromosome 1, Rhd and Rhce. The Rhd gene specifically codes for the Rhd protein, which expresses the D antigen. Its presence confers Rh positivity, while its absence means negativity.
Simple enough for D.
For D, yes. The Rhce gene, on the other hand, codes for the Rhce proteins, which express the CC and E antigens. The combination of these two genes determines 8 main haplotype-specific combinations of these alleles on the chromosome, like DC, DCE, DCE, and their D, absence of D, counterparts like DC, DCE, etc. The frequencies of these haplotypes vary considerably among different geographical populations.
And what about the less common expressions, things like weak RHD or partial RHD? How are those identified, and why is it so vital to distinguish them accurately?
That's a really important point. The Rh antigen diversity includes instances of weak or partial expression, where the D antigen is present but in a modified or reduced form on the red cell surface. Identifying individuals with weak or partial RHD is absolutely vital for several reasons.
Why is it so critical?
Especially for young females of childbearing potential, to ensure appropriate Rhe negative blood is given, if needed, to prevent them from making anti-D antibodies, which could harm future pregnancies, causing HDFN. It's also crucial for multi-transfused patients to ensure appropriate blood selection and prevent antibody formation. Historically, distinguishing these variants was challenging with just serological antibody-based methods, but molecular biology, DNA testing basically, is now routinely used to accurately resolve such situations by directly testing the underlying genetic variations. It provides precise identification.
Next up in the table, the KEL system. What's its significance in transfusion medicine, and is there anything unique or particularly interesting about it?
The KEL system has 36 antigens, but typically only the K antigen, KEL1, is routinely determined in most transfusion services. Why? Because of its high immunogenicity.
Meaning it easily provokes an immune response.
Exactly. Its immunogenicity ranks third after ABO and RH, making it clinically very significant for transfusion safety. Approximately 9% of Caucasians and 2% of individuals of African ancestry are K positive.
Okay. Anything else notable?
Yes, what's particularly fascinating and unique about the KEL system is its connection to the Kerix protein. The rare absence of this protein, which is controlled by an X-linked gene, leads to a specific weak Ke antigen expression. But more dramatically, it results in abnormal red blood cells called acanthocytes, shortened red blood cell survival, and a progressive form of muscular dystrophy that can include serious cardiac defects.
Wow, a blood group linked to muscular dystrophy.
Yes. This rare condition is known as the McLeod phenotype. It showcases a truly surprising and profound link between a seemingly obscure blood group system and a severe systemic disease. It underscores just how deeply intertwined these seemingly distinct parts of our biology can be.
That's truly astounding. It really underscores just how deeply interconnected these seemingly distinct parts of our biology are. It makes you wonder what other connections are hiding in plain sight. Let's move to the Duffy system.
Okay, the Duffy system, FY, comprises 5 antigens. Of these, FIA, FY1, and Phoebe, FY2, are the 2 routinely tested antigens, encoded by their respective FIA and Phoebe alleles. This leads to 3 common phenotypes, Phi A plus B plus, Phi A plus B, and Phi AB plus, depending on the combination of alleles and individual inheritance.
And there's a unique phenotype within the Duffy system, one that confers natural resistance to a specific type of malaria, isn't there? That's a powerful evolutionary connection.
Yes, that's a very important and clinical powerful point for the Duffy system. The Phi AB phenotype, characterized by the complete absence of both Phi and Phi antigens, is almost exclusively found in individuals of African ancestry. Its frequency there can reach an astonishing 70–100%, depending on the specific population.
Why is that significant?
This distribution is highly significant because the Duffy antigens serve as crucial receptors for Plasmodium vivax, one of the main malaria parasites.
So no Duffy antigen, no entry for the parasite.
Exactly. The Phi AB phenotype confers a profound natural resistance to P. vivax malaria, providing a clear evolutionary advantage in regions where malaria is endemic. However, these individuals can still develop antibodies against 2 other high-frequency Duffy antigens, FY3 and FY5, after transfusion or pregnancy, complicating future transfusions. They may also present with low granulocyte counts, incidentally, but this is generally not associated with disease.
A clear evolutionary advantage written in blood groups. Amazing. How about the KID system?
The KID system, JK, has 3 antigens, with JK, JK1, and JKB, JK2 being the 2 routinely tested ones, encoded by their respective JK and JKB alleles. Like Duffy, it typically results in 3 common phenotypes, JKA plus a B plus, JKA plus B, and JKAB plus.
Is there a null phenotype here too?
Yes. There's also a particular rare phenotype known as JKAB, characterized by the complete absence of both JK and JKB antigens. This rare phenotype is found predominantly in Polynesian populations and is linked to a silent JK0 allele. Individuals with this phenotype may develop antibodies against the high-frequency anti-JK3 antigen after transfusion or pregnancy, which can make it difficult to find compatible blood later on.
Finally, let's unpack the MNS system. It sounds quite extensive with 49 antigens mentioned. What's important for us to know about it?
The MNS system is indeed quite extensive, comprising 49 distinct antigens, although typically only 4 are routinely tested. 2 genes, GYPA and GYPB, encode 2 pairs of antithetical antigens.
Antithetical opposites.
Yes, basically opposites, where the presence of one typically implies the absence of the other. The M, MNS1, and N, MNS2 antigens are expressed on a protein called glycophorin A, while S, MNS3, and MNS4 are expressed on glycophorin B. The specific combination of these alleles determines their presence. For example, an M plus N plus subject would be homozygous for the MNS alleles.
Okay. Any rare phenotypes here to worry about?
Yes, similar to the other systems, a rare phenotype, NSSU, exists. This particular phenotype is found primarily in individuals of African ancestry. These individuals can develop a potent antibody against the high-frequency U antigen, MNS.5, after transfusion or pregnancy, again posing significant challenges for finding compatible blood for future needs. 2.5 rare RBC phenotypes and transfusion challenges.
Given all this incredible diversity, I mean, hundreds of antigens across dozens of systems, it sounds like finding truly compatible blood can be an incredibly challenging, almost Herculean task sometimes, especially for certain patients or in specific populations.
It absolutely is. And it's where the true art and science of transfusion medicine profoundly intersect. Some patients present with extremely rare genotype-to-phenotype assortments. This means their red blood cells might display so-called private antigens, which are truly unique to them or a very small group.
Or they might be missing common ones.
Exactly. Conversely, they might entirely lack public antigens, which are antigens that are widely shared across the general population. If a patient becomes public negative immunized, meaning they lack a common antigen and have developed antibodies against it after prior exposure, transfusing them using conventional blood bank resources becomes virtually impossible. It's a genuine medical emergency. Such complex cases demand immediate access to designated blood banks with specialized rare blood programs. Their primary, often life-saving role is precisely to identify, recruit, and collect blood from donors who exhibit these uncommon antigen displays. It requires a global network and meticulous record keeping.
So specific ethnic populations might even be targeted for donations because of their unique genetic profiles and the higher prevalence of certain rare phenotypes within those groups. It's almost like a precision search for donors.
Precisely. This is a crucial and ethically sensitive aspect of managing rare blood needs, yet it's entirely focused on patient safety and availability. Some ethnic populations indeed display genotype specificities that lead to a higher prevalence of certain rare blood types. For instance, the Bombay group, a very rare and incredibly challenging blood type, is found more frequently in southwestern Indians. Similarly, conditions like sickle cell disease, which is more common in individuals of African ancestry, often involve RBC phenotypes that are uncommon in Northern Hemisphere countries. This creates significant challenges in matching donors for these patients, as finding compatible blood within local populations can be difficult.
So what do they do?
In these situations, it sometimes necessitates the use of highly valued cryopreserved blood components, that frozen blood we talked about, as a last resort when immediate fresh matches simply cannot be found. This raises an important question. How do we manage these profound biological complexities in everyday clinical practice, ensuring that every patient receives the best possible care despite these intricate biological hurdles and rare genetic profiles?
Clinical indications and efficacy. The art and science of when and how to transfuse.
So now that we understand the components themselves and the intricate biology of matching, let's pivot to the clinical applications. Transfusions are undeniably life-saving, but our source also calls them scarce resources with potential adverse reactions. What's the guiding philosophy here for when and how to transfuse? How do clinicians make these critical decisions in practice?
This is where patient blood management (PBM) programs truly come into their own. They represent a significant and transformative shift in thinking in transfusion medicine. They emphasize a multidisciplinary approach, recognizing that transfusion isn't a standalone solution, but rather one tool within a broader holistic strategy for managing a patient's blood.
A broader strategy. How so?
The PBM philosophy integrates three key proactive pillars. First, optimizing hematopoiesis, meaning proactively enhancing the body's own ability to produce blood cells before a need arises, like treating underlying deficiencies.
Right.
Second, meticulously minimizing blood loss during interventions like surgery through careful surgical techniques, advanced hemostatic agents, and pharmacological agents.
Stop the bleeding before it starts, essentially.
Exactly. And third, crucially, optimizing a patient's intrinsic tolerance to anemia, allowing the body to adapt to lower hemoglobin levels where it is clinically safe to do so, avoiding unnecessary transfusions based just on a number.
So it's about making sure transfusions are truly necessary, are used as efficiently as possible, and that all viable alternatives are robustly considered first. It sounds like a conservative but deeply patient-focused approach.
Exactly. It's about judiciously using a precious and potentially risky resource while putting the patient's overall well-being and physiological reserves at the forefront. 3.2 specific blood component uses and efficacy, Table 113-4, deep dive.
Table 113-4 from our source is incredibly detailed, acting like a clinician's precise roadmap. It lays out the exact therapeutic indications, the goals, compatibility requirements, dosages, and how we evaluate the efficacy for each blood component. Let's break this down thoroughly for our listeners, starting with the most common and often first thought of. Red blood cell concentrates, or RBCCs.
Certainly, this table is packed with essential clinical detail for understanding transfusion practice. Let's start with red blood cell concentrates, RBCCs. Their primary therapeutic indication is for anemia and/or tissue ischemia, aiming for both treatment and prevention of inadequate oxygen supply to tissues. The fundamental goal is straightforward, to improve systemic and tissue oxygenation.
Getting oxygen to the tissues.
Exactly. Now, thresholds for transfusion. It's vital to understand that it's not just about a single hemoglobin number. Clinical symptoms of anemia and oxygen deprivation are absolutely crucial. Generally, the accepted hemoglobin threshold is below 7 GDL for hemodynamically stable patients.
Okay, 7 grams.
But this threshold rises. It's below 8 GDL for patients undergoing orthopedic or cardiac surgery, or those with pre-existing cardiovascular disease, as they have higher oxygen demands and less reserve.
Right.
For patients with acute coronary syndrome, it's even higher, typically below 9-10 GDL, due to the critical need for cardiac oxygenation. It's crucial to note, however, that these specific thresholds do not apply to neonates or to patients with severe thrombocytopenia or chronic transfusion-dependent anemia. They have different needs.
And when not to use them.
Critically, RBCCs are not recommended for nutritional anemia like iron, B12, or folate deficiency. Those should be treated by addressing the underlying deficiency, not just replacing cells temporarily.
Makes sense. Compatibility.
For RBCCs, ABO-compatible cellular and ideally ABO-identical blood is always preferred. RHD compatibility is a strict requirement for young and childbearing females and whenever possible for multi-transfused patients. For frequently transfused patients, matching for RHCE and KEL is also needed. Addition matching might be required based on antibody screens. Typically, 1 unit of RBCC is about 250-350 millis, is transfused at a time, repeated based on clinical status and HB response.
And how do you know it worked?
Efficacy is evaluated by a tangible reduction of anemia-related symptoms, overall clinical improvement, and a measurable increase of hemoglobin, usually about +1 GDL per unit, and hematocrit about +3%. Host speaker. Okay, what about RBC exchange? That sounds different. It is. It's a more specialized procedure. Therapeutic indication. Primarily used for managing anemia or severe sickle cell crisis in patients with hemoglobinopathies like sickle cell disease or thalassemia. Goal: to proactively replace the altered or sickled red blood cells with healthy donor cells, compensate for hemolysis, and prevent severe sickle cell crises. Dosage: typically 25-30 millilograms of patient body weight of red cells are exchanged. Efficacy evaluation. For sickle cell, efficacy is primarily assessed by a measurable reduction in the percentage of HbS, the abnormal hemoglobin, to a safer level. Host speaker. Got it. Next up, platelet concentrates, PCs for clotting, right, vital for hemostasis. Therapeutic indication. Specifically for thrombocytopenia-related bleeding disorders when platelet counts are too low, used for both treating active bleeding and for protecting bleeding. Thresholds. Platelet levels are key. Generally 5,000 Ls without fever infection, a 10,000, 20,000 Ls for fever infection, 50,000 Ls for surgery, DIC, or invasive procedures, and even higher, 80,000 Ls for neurosurgery or eye surgery, where even tiny bleeds are catastrophic. They're also used in massive bleeding scenarios, not recommended for. Importantly, PCs are generally not recommended for immune causes of low platelets like ITP, TTP, or HIT, as the transfused platelets would likely be destroyed just as quickly. Goal: to correct impaired primary hemostasis and promote vessel healing. Fascinating detail. Our source highlights that cold-stored platelets, despite surviving less time in the body than room temp platelets, surprisingly maintain and possibly even improve their hemostatic capacity. This is a hot area of research. Compatibility. ABO-identical PCs are best. If not, ABO-compatible, cellular, with low-titer anti-AB antibodies. RHC-compatible, preferred in premenopausal women. HLA-compatible or identical PCs for patients refractory due to anti-HLA antibodies. HPA-compatible for specific neonatal conditions. Dosage. Typically 0.50-0.7 by 710 platelets per efficacy evaluation, primarily preventing or stopping bleeding. Quantitatively, the corrected count increment, CCI, is used, aiming for 010, 109 L within 1 hour, though this isn't applicable to cold or cryopreserved platelets. Host speaker. Okay, moving on to plasma. What's its role? Plasma contains clotting factors, therapeutic indication, primarily for coagulation, factor-related bleeding disorders, and acute hypovolemic coagulopathy where factors are depleted. Experimentally, it's been used for infections like convalescent plasma. Goal: to correct impaired hemostasis by providing missing coagulation elements or factors for vessel healing, or provide specific antibodies. Compatibility. ABO-compatible but based on the plasma, not the cells. Dosage, generally 10-15 ml/kJ. Efficacy evaluation. reduced bleeding or maybe infection resolution, host speaker. And plasma exchange, that's removing plasma. Yes, a therapeutic apheresis procedure. Therapeutic indication, used for rapid removal of pathogenic antibodies or toxins, or supplementing missing enzymes, like in PTP or Guillain-Barré. Also used to remove anti-HLA antibodies before kidney transplant. Goal, to deplete harmful elements like autoantibodies or excess cholesterol. The replacement plasma can also bring beneficial anti-inflammatory factors. Compatibility, ABO-compatible plasma. Dosage, typically 45-60 millicovirag exchanged. Efficacy evaluation, improve disease symptoms like platelet recovery in TTP, or reduce antibody levels before transplant. Host speaker, what about whole blood? Is that still used much? Less common for routine use now, but still has a role. Therapeutic indication, primarily for acute hypovolemic coagulopathy requiring massive transfusion, like severe trauma. Goal, to provide a balanced provision of all blood components, maintained at 4 degrees C, without dilution from additive solutions. Crucial in massive blood loss. Compatibility, ABOI identical or group O with low titer anti-AB antibodies. Dosage, repeated based on clinical status. Efficacy evaluation, restoring blood volume, normal volemia, and stopping bleeding. Host speaker, so instead of whole blood, multi-component therapy is more common now for massive bleeds. Yes, often preferred as it allows more flexibility. Therapeutic indication, acute hypovolemic coagulopathy needing massive transfusion. Mimics whole blood, but allows adjustments. Goal, provide components in an appropriate ratio reflecting needs during massive loss. A ratio of 1 RBCC 1 plasma 0.25 PC is currently favored, though research continues. Compatibility, standard rules apply for each component given. Dosage, administered in that favored ratio, repeated as needed. Efficacy evaluation, normal volemia, and bleeding resolution. Host speaker. Okay, now granulocyte concentrates. These sound very specialized. They are, and somewhat debated. Therapeutic indication, for severe refractory bacterial or fungal infections in patients with profound neutropenia, very low white cell count 100L, or with dysfunctional granulocytes, like in CGD. Important note, their usefulness is still debated. Formal proof of efficacy is lacking for many situations, often a last resort. Goal, to correct impaired granulocyte function. Compatibility, ABO compatible. Dosage, typically 1 to 2 of 1,010 granulocytes repeated. Efficacy evaluation, infection resolution or stabilization until the patient's marrow recovers. Important process detail. Collecting these requires giving the donor steroids or growth factors beforehand, and they're exposed to heparin and HES during collection. Not insignificant for the donor. Host speaker. Donor mononuclear cells, that sounds like immunotherapy. Essentially, yes. Used in specific cancer contexts. Therapeutic indication, primarily for relapse of blood cancers after allogeneic hematopoietic cell transplant. Goal, to induce a powerful graft versus leukemia effect where donor immune cells attack remaining cancer cells. Also enhances the graft. Dosage, T lymphocyte count is carefully adjusted, maybe 105, 107 T cells clock shape. Very precise. Efficacy evaluation, disease specific, looking for remission. Host speaker. And lastly, cryoprecipitate. What's in that? It's a concentrate of specific clotting factors. Therapeutic indication, used for acute bleeding, specific types of von Willebrand disease, and hemophilia A if factor concentrates are unavailable. Goal, to provide concentrated fibrinogen, factor 8, von Willebrand factor, and factor 13th, all key for clotting. Compatibility, ABO compatibility generally not required. It's about the proteins. Dosage, small volume per unit, usually pooled, 4 to 5 units. Efficacy evaluation, primarily assessed by an increased plasma fibrinogen level.
That's an incredibly comprehensive breakdown. It's really clear that the decision to transfuse, and with what specific component, is a highly nuanced and evidence-driven process demanding real expertise.
Navigating the risks, adverse transfusion reactions.
So we've established that transfusions are undeniably life-saving, but they also come with inherent, often complex risks. What's the general picture regarding these adverse reactions, and how are clinicians trained to spot and manage them quickly?
Right. While indispensable, transfusions do carry a spectrum of risks. Most commonly, adverse reactions are non-life-threatening and relatively mild. But a critical point emphasized by our source is that severe reactions can sometimes present with mild initial symptoms.
You can't dismiss mild symptoms.
Exactly. This underscores the absolute continuous need for close monitoring of transfused patients for any warning signs, as detailed in Table 113-5. When a reaction is suspected, the immediate non-negotiable step is to stop the transfusion immediately, then rapidly assess the recipient's clinical status and initiate supportive care as needed.
Okay. What does the data say about fatalities?
Looking at recent U.S. data, between 2014 and 2018, approximately 35 transfusion-associated fatalities were reported yearly to the FDA. That's among about 14 million blood components transfused.
Very rare, relatively speaking.
Very rare, which reflects the extensive safety measures. But it highlights that severe risks persist. The most frequent causes of death during that period were transfusion-associated circulatory overload, TICO, at 32%, followed by trachea, 26%, hemolysis, 18%, and sepsis, 14%.
That's a powerful and perhaps unexpected twist. After all the breakthroughs in preventing complex immune reactions and infections, it's actually fluid overload TICO, something that might seem more straightforward, that has emerged as the leading cause of transfusion-related fatalities now. That fundamentally reshapes what clinicians need to be most vigilant about. What are the broad categories of causes for these reactions? How do they originate?
Precisely, it's a critical shift in understanding risk. Reactions generally stem from two broad categories of mechanisms, either immune-mediated mechanisms or non-immune mechanisms. Immune-mediated reactions are typically due to recipient or donor alloimmunisation, that antibody formation or performed antibodies. Non-immune causes arise from the physical or chemical properties of the blood components, or, critically, from pathogens within the component. 4.2 Immune-mediated adverse reactions, unpacking the body's responses.
Let's start with the most dramatic and potentially life-threatening hemolytic reactions. What exactly happens in acute hemolysis, and why is it so dangerous and rapid when it occurs?
Immune-mediated acute hemolysis is a true medical emergency. It occurs when the recipient's preformed antibodies immediately or very rapidly, during or within 24 hours of transfusion leaves, or violently destroy the transfused donor red blood cells.
Immediate destruction.
Yes. The most severe, often fatal reactions are typically caused by anti-A or anti-B antibodies due to their potent ability to activate the entire complement system. However, alloantibodies against other red blood cell antigens like Rh, Cal, or Duffy can also cause severe hemolytic reactions, though sometimes with a slightly different tempo or mechanism.
What typically causes these severe reactions, particularly given all the layers of safeguards like cross-matching? Is it usually a failure of the immune system itself or something more, well, preventable?
Alarmingly and tragically, these dramatic reactions are usually caused by critical errors in the transfusion process.
Human error.
Often, yes. This includes fundamental failures in product or patient identification, giving the wrong blood to the wrong patient, or erroneous blood grouping in the lab, or missing an unidentified pre-existing anti-RBC along an antibody in the recipient. Less severe hemolysis can also occur from transfusing components containing incompatible plasma with lots of antibodies against the recipient's RBCs. Typically seen with platelet transfusions, the estimated frequencies are low, 110 per 100,000 for acute reactions, maybe 540 per 100,000 for delayed ones, but the risk is real.
The source provides Figure 113.1, which offers a detailed illustration of the complex mechanisms behind these hemolytic reactions. Can you walk us through those different crucial pathways? How does the destruction actually happen?
Yes, Figure 113.1 is very instructive. It illustrates three main mechanisms. First, A. Acute intravascular hemolysis. This is the most severe immediate type. Pre-existing antibodies, like natural anti-B or IgG from prior sensitization, react with antigens on the transfused RBCs. This triggers the complement cascade like dominoes falling. It activates all the way to C5-C9, forming the membrane attack complex.
The MSE attack.
Right. Think of it as a drill creating pores in the RBCs, leading to their destruction within the blood vessels, intravascular hemolysis. This is extremely dangerous. It releases toxic free hemoglobin, causing kidney failure, and can trigger DIC, that widespread clotting and bleeding. Host speaker. Okay, what's the second mechanism? B, extravascular hemolysis, acute or delayed. Often seen in delayed reactions, usually involving newly formed IgG antibodies. Here, complement activation is incomplete, stopping around C3. This leads to C3b-mediated opsonization. The RBCs get tagged with C3d.
Tagged for destruction.
Essentially, yes. These tagged RBCs are then cleared via immunophagocytosis by macrophages, mainly in the spleen and liver, outside the blood vessels. The main signs are anemia and jaundice, less dramatic than the shock of intravascular hemolysis. Host speaker. And the third, C, antibody-dependent cellular cytotoxicity, ADCC, mediated phagocytosis. Here, alloantibodies bind to the RBCs but without activating complement. Instead, these antibody-coated RBCs are directly recognized by macrophages via their SCSC receptors, leading to engulfment and destruction.
So, given how potentially life-threatening these reactions can be, especially the intravascular kind, how do we prevent them in the first place? What are the critical steps taken before a transfusion even begins?
Prevention relies heavily on meticulous pre-transfusion testing. This includes accurately determining the patient's ABO and RHD phenotype and identifying any pre-existing anti-ABO antibodies. Depending on the context, additional typing for other Rh antigens, CCEE, KEL, K, and sometimes Duffy, KID, and S's might be done. While traditional serology is common, molecular typing DNA testing is increasingly used to predict the phenotype accurately, especially in complex cases.
Beyond the lab tests.
Crucially, prevention extends to rigorous procedural checks. Meticulous patient identity verification at every single step, from drawing the sample to hanging the bag, and adequate tube labeling are paramount to prevent those clerical errors. A double ABO determination, using 2 separate samples, is often considered best practice, especially without a systematic cross-match.
What about detecting those irregular antibodies you mentioned earlier, the ones that aren't ABO or RHD? How are those identified, and why is that so important?
Testing also includes a vital step: screening and identifying allo antibodies against RBC antigens other than ABO. This is done by mixing the patient's serum with selected typo RBCs known to express various common antigens. If agglutination occurs, the antibody's specificity is identified by correlating the reaction pattern.
Like a puzzle.
Exactly. It's important to note some drugs like anti-CD38 for myeloma can interfere with this screening, but labs have ways to manage that, like DTT pretreatment. Finally, cross-matching between the recipient's plasma and a sample from the selected donor unit is often performed as a final check, especially for alloimmunized or frequently transfused patients, or in settings like sickle cell disease. It's that final layer of safety.
So if a patient is D negative, we always give them D negative blood, right, to avoid any potential RH issues.
Ideally, yes, that is the gold standard. For Rh1 negative patients, every effort is made to provide Rh negative blood components to prevent anti-D alloimmunization. However, in true emergencies where D negative blood isn't available and life is at stake, D positive RBCCs can be transfused to a D negative patient lacking anti-D antibodies.
But there's a risk.
Yes, a significant risk.
Yeah.
An estimated 22% of those recipients will become alloimmunized and produce anti-D antibodies after getting D positive RBCs. The risk is much lower, maybe 1%, with platelet transfusions.
So it's a calculated risk in emergencies.
It is, and a truly critical recommendation here. Whenever possible, females of childbearing potential, including even young girls, should always receive TEL1 compatible RBCCs and D-compatible PCs. This is paramount to prevent alloimmunization that could lead to severe HDFN in future pregnancies.
You mentioned minor ABO incompatibility earlier, especially with platelet concentrates, which seems counterintuitive if the cells match. Could you elaborate on how that leads to problems?
Yes, it's a subtle but important point. ABO plasmatic incompatibility, or minor incompatibility, occurs when donor antibodies in the plasma of the blood component target the recipient's red blood cell antigens. While RBCCs have minimal plasma, platelet concentrates contain 100-300 milliliter of plasma. If these PCs come from a donor with high-titer anti-A or anti-B antibodies...
High levels of antibodies.
Right. Those antibodies can induce hemolytic reactions in the recipient, even if the recipient's red cells are ABO compatible with the donor's red cells. To mitigate this, ABO-identical PCs are preferred. If not possible, PCs from donors with only low-titer anti-AB should be selected. High-titer unit should generally be restricted to Group O recipients. Platelet additive solutions significantly reduce this risk by lowering plasma volume. Additionally, this incompatibility can form immune complexes, leading to inflammation and platelet activation.
What are the classic observable symptoms that would make a clinician immediately suspect an acute hemolytic reaction is occurring during a transfusion? What are the red flags they look for?
They can present dramatically and rapidly. Classic symptoms include sudden hypotension, a sharp drop in blood pressure, tachypnea, rapid breathing, tachycardia, rapid heart rate, fever rising 1-2 degrees C, severe chills, and patient complaints of chest and back pain. More specific and alarming signs are hemoglobinuria, dark, cola-colored urine from free hemoglobin, and hemoglobinemia, free hemoglobin in the blood plasma. In severe cases, this can escalate quickly to DIC, acute renal failure, profound shock, and potentially death. It's a race against time.
And what about delayed reactions? How do they manifest, and why are they delayed, sometimes appearing weeks later?
Delayed hemolytic reactions primarily manifest as persistent or worsening anemia, often accompanied by icterus or jaundice. These typically occur 1-2 weeks after the transfusion. They result from an anamnestic response.
A memory response.
Exactly, a secondary immune response. This happens in previously sensitized patients whose antibody levels were too low to detect before transfusion. The transfusion acts as a re-exposure, triggering a rapid, amplified antibody production. The alloantibody then becomes detectable, and red cell destruction occurs, usually 1-2 weeks post-transfusion.
How is hemolysis ultimately diagnosed in the lab to confirm the clinical suspicion and identify the specific antibody involved?
Diagnosis relies on clinical signs plus specific lab findings: persistent anemia, depleted plasma haptoglobin, which binds free hemoglobin, hemoglobinemia, hemoglobinuria, and elevated LDH in an unconjugated bilirubin, all markers of red cell destruction.
And the Coombs test.
Yes, the direct antiglobulin test, BAT, or direct Coombs test, is usually positive. It detects immunoglobulin or complement coating the recipient's RBC surface, indicating an immune attack. Figure 113-2 shows this. Similarly, the indirect antiglobulin test, IAT, or indirect Coombs test, which detects anti-RDC alloantibodies in the serum, will also be positive, also in figure 113-2. An elution procedure can then remove antibodies from the RBC surface to identify the specific culprit alloantibody.
Once an acute hemolytic reaction is diagnosed, what's the immediate and ongoing management strategy? It sounds like time is absolutely critical.
Management is primarily supportive, focusing on preventing further harm and managing complications. Critical initial steps: promptly stop the transfusion, perform a biological workup, and conduct a thorough clerical check to prevent a second error for the patient. Vigorous hydration with isotonic saline and diuretics to maintain urine output and protect the kidneys. Severe cases might need forced alkaline diuresis, electrolyte correction, pressor support for shock. If DIC and severe bleeding occur, PCs, plasma, and cryoprecipitate may be needed. In rare, unavoidable situations of transfusing incompatible RBCs, prophylaxis with steroids and IVI has been used to minimize hemolysis.
Can immune-mediated hemolysis also occur in other contexts, maybe after a transplantation, even without a direct blood transfusion? That seems like a surprising connection.
Yes, it absolutely can. Immune-mediated hemolysis can occur after allogeneic hematopoietic transplantation, less common with solid organs. The most common cause here is minor ABO incompatibility, where viable donor B lymphocytes, passenger lymphocytes, transferred with the graft produce alloantibodies targeting recipient red cells, for example, anti-D from a D-negative donor into a D-positive recipient. This typically happens 5-14 days post-transplant. Reduced intensity conditioning and cyclosporine might increase risk. Prevention involves transfusing RBCs compatible with the donor and using GVHD prophylaxis targeting B cells.
And major incompatibility in transplant.
Major ABO incompatibility, recipient antibodies destroying donor-derived red cells, can also cause acute hemolysis or prolonged red cell aplasia. Removing red cells from the graft beforehand, deserythrocytation, can reduce early risk.
And what about polyvalent immunoglobulin itself, the therapeutic product given to patients? Can that cause hemolysis? Seems ironic.
Surprisingly, yes. Polyvalent immunoglobulin, IVA, can contain high titers of anti-A and/or anti-B antibodies, potentially inducing acute hemolysis, though usually mild. This is particularly seen in Group A or AB children getting high-dose IVA for things like Kawasaki's disease, or adults treated for TTP. A similar mechanism can cause hemolysis after anti-D immunoglobulin treatment for ITP in Rhd-positive patients.
Are there non-immune causes of hemolysis as well? Things not related to antibodies attacking red cells, mechanical damage maybe?
Absolutely. Non-immune mechanisms exist. These include thermal, overheated cold components, osmotic, concurrent hypoosmotic fluids causing cells to burst, and mechanical pressure from high-flow devices or cell saver processing physically damaging cells.
Can other underlying medical conditions complicate the picture, making it difficult to distinguish from a true transfusion reaction? It sounds like a diagnostic puzzle sometimes.
Absolutely, it can be challenging. Autoimmune and drug-induced hemolytic anemias can be exacerbated by transfusion, mimicking HTRs. Transfusing RBCs with enzymatic defects, making them fragile, can also mimic immune hemolysis.
And hyperhemolysis.
Yes, a severe and concerning reaction is hyperhemolytic transfusion reaction, especially in patients with hemoglobinopathies like sickle cell disease on long-term transfusions. A transfusion can trigger bystander hemolysis, where the patient's own red cells are destroyed along with the transfused ones. It's thought to involve a systemic inflammatory response leading to macrophage lysis of red cell precursors. It can be immediate or delayed, causing life-threatening drops in hemoglobin, often below pre-transfusion levels. Critically, further RBCC transfusions usually worsen the ongoing hemolysis. Management is incredibly difficult. 4.2.2 febrile non-hemolytic transfusion reaction, FNHTR.
So if acute hemolysis is the most traumatic, what's the most frequent adverse reaction associated with transfusions? It sounds like there's a More common but less dangerous, one clinicians see often.
That would be the febrile non-hemolytic transfusion reaction, FNHTR. And yes, it is indeed the most commonly reported reaction, typically associated with cellular blood components like red cells or platelets. It's characterized clinically by chills, rigors, and a rise in body temperature of at least 1 degree C, usually within 4 hours of transfusion. While uncomfortable, it's generally not life-threatening.
What causes it? If it's not hemolysis breaking down cells, what's the mechanism behind this frequent reaction?
It's caused by one of two primary mechanisms, either by pro-inflammatory cytokines that accumulate within the blood component itself during storage.
Chemicals released by the cells.
Exactly. Or by recipient antibodies directed against donor cell antigens, most often HLA antigens on white cells present in the blood component, leading to a mild immune activation. It's crucial to remember that FNHTR is a diagnosis of exclusion. Other, more serious causes of fever, notably bacterial infection and acute hemolysis, must be thoroughly ruled out first.
And how is it prevented? Are there effective strategies to reduce its incidence, given how common it is?
Yes, very effective strategies exist. Leukocyte reduction, especially press storage reduction, is highly effective in preventing FNHTRs by removing the cells that release those pro-inflammatory cytokines. Also, the use of additive solutions for platelets decreases its frequency. Interestingly, pre-medication with antipyretics like acetaminophen has generally proven ineffective at reducing the rate of these reactions and, importantly, may actually mask symptoms of a more serious reaction. So prevention through leukoreduction is key. 4.2.3, allergic reactions.
Allergic reactions are also on the list of potential adverse events. How do they typically present, ranging from just mild, itchy rashes to the most severe, life-threatening forms like anaphylaxis?
Most allergic transfusion reactions are relatively mild, presenting as self-limiting symptoms like rash, pruritus, itching, urticaria, hives, or localized edema. These are the most common types.
Okay, hives and itching.
Right. More rarely, however, reactions can escalate to severe or even life-threatening anaphylaxis. This involves dramatic symptoms like bronchospasm, airway narrowing, respiratory distress, profound hypotension, nausea, vomiting, and shock. Mild reactions occur maybe 1,000 times per 100,000 components, while severe anaphylactic ones are much rarer, 110 per 100,000.
How are they managed?
Mild reactions can often be managed by temporarily stopping the transfusion and giving antihistamines. For patients with a history of allergic reactions, pre-medication with antihistamines is sometimes used, though there's no universal consensus on its effectiveness. For extremely sensitized patients, cellular components can even be washed to remove residual plasma and potential allergens.
What's the underlying mechanism for these allergic reactions, particularly the severe ones like anaphylaxis? What triggers such a dramatic response?
Most mild allergic presentations may not depend on specific preformed antibodies. Instead, they're often attributed to soluble mediators present in the blood component or generated during transfusion. These mediators can trigger widespread release of histamine and serotonin from the recipient's platelets and leukocytes, causing the allergic symptoms.
And anaphylaxis.
An anaphylactic reaction can occur after just a few milliliters of blood. Treatment involves immediately stopping the transfusion, maintaining IV access, and promptly giving adrenaline, epinephrine. Additional steroids, antihistamines, and bronchodilators might be needed.
Is there a specific patient population at higher risk for severe allergic reactions, beyond just a general history of allergies? Something specific in their biology?
Yes, there is. Individuals with IgA deficiency, affecting less than 1% of the population, may be sensitized to this IgA immunoglobulin isotype. If they are, they face a heightened risk of anaphylactic reactions when receiving plasma transfusion, as donor plasma contains IgA.
So they react to the IgA itself?
Potentially, yes. As a precaution, severely IgA-deficient individuals should, if available, receive IgA-deficient plasma and washed cellular components. Patients with anaphylactic or repeated allergic reactions should be tested for IgA deficiency. However, the exact clinical importance of this specific risk is currently debated, with some studies questioning its overall significance. 4.2.4 graft versus host disease, GVHD.
GVHD is well known as a major complication in organ and bone marrow transplant, but it can happen from regular blood transfusions too. That sounds incredibly serious.
It is an extremely rare adverse reaction from transfusion, thankfully, though much more frequent after allogeneic hematopoietic transplant. Transfusion-related GVHD is mediated by engrafted donor T lymphocytes inadvertently transfused with blood components. These T cells then proliferate and mount an immune response in a recipient who is profoundly unable to reject these foreign lymphocytes.
Why can't the recipient reject them?
either due to severe immunosuppression in the recipient or more subtly because the recipient shares an HLA haplotype with the donor, so the donor lymphocytes aren't fully recognized as foreign and escape rejection.
How does it manifest clinically, and what's its prognosis? It sounds terrifying.
These donor T lymphocytes, once engrafted, react against the host's HLA antigens. Clinically, it typically develops 5-10 days after transfusion. It presents with systemic immune activation, cytopenia, low blood counts, fever, a characteristic diffuse skin rash, severe diarrhea, and liver function abnormalities. What's truly alarming is the prognosis. Transfusion-associated GVHD is highly resistant to standard immunosuppressive therapies, and it's fatal in over 90% of cases. It's genuinely one of the most feared transfusion complications.
How do we prevent something so devastating, given its rarity but such severe outcomes? What are the key preventative measures?
Prevention in at-risk patients relies critically on irradiation of cellular blood components, delivering a minimum dose of 25J, or treating components with pathogen reduction technology that depletes living cells, including T lymphocytes.
Who are the at-risk patients?
Those with inherited immune deficiencies, patients undergoing hematopoietic transplant, autologous or allogeneic, patients on specific potent immunosuppressive drugs, like purine analogs, anti-CD52-AB, ATG, fetuses receiving intrauterine transfusions, and recipients of blood from a blood relative, higher chance of HLA sharing. Because granulocyte concentrates contain many lymphocytes, they must always be irradiated. 4.2.5 transfusion-related acute lung injury, TARI-LI.
TARI-LI is another serious one, often mentioned as a major cause of transfusion-related fatalities. What defines it clinically, and how quickly does it typically occur after a transfusion starts?
TARI-LI is characterized by the sudden occurrence or significant worsening of hypoxia, low blood oxygen, and non-cardiogenic pulmonary edema fluid in the lungs not caused by heart failure. This shows up as bilateral interstitial infiltrates on chest X-ray. It classically occurs during or within 6 hours after transfusion, though delayed cases up to 72 hours are seen.
How common is it?
Frequency ranges from 0.5 to 10 per 100,000 components. It's often hard to distinguish from other causes of acute hypoxia, especially circulatory overload, ATACO, but it is among the most common causes of transfusion-related fatalities. Treatment is unfortunately supportive only oxygen, ventilation support.
So what causes TLI? What's the immune mechanism at play here, and why is it so devastating to the lungs specifically?
TLI usually results from the transfusion of donor plasma containing high-titer anti-HLA class Ti antibodies that bind to the recipient's white blood cells. Anti-HLA class I and anti-human neutrophil antigen, HNA, antibodies can also be involved.
So it's antibodies again.
Often, yes. But TLI can also be mediated by cytokines and chemokines without a direct antibody interaction. In these cases, leukocytes, especially if primed by bacterial components or inflammation in the patient, aggregate abnormally in the lungs' blood vessels. They then release inflammatory mediators, causing severe acute lung injury. The lungs essentially get flooded.
How has the risk of TLI been so significantly reduced, given its severity and past frequency? It sounds like there was a major shift in blood banking practice.
You're right, there was a major impactful policy shift. The risk has been significantly reduced where policies have been implemented to preferentially use plasma and platelet concentrates from male donors and nulliparous, never pregnant, or paris, previously pregnant, female donors without anti-HLA antibodies.
Why those donors specifically?
Because pregnancy is a major way women can become sensitized to HLA antigens, leading to the formation of these antibodies, identified as a primary cause of TLI. Recipient risk factors for TLI include smoking, chronic alcohol use, shock, liver surgery, cancer surgery, and sepsis. 4.2.6, post-transfusion purpura, PTP.
Post-transfusion purpura sounds like it specifically affects platelets, given the name purpura often refers to bruising from low platelets.
Yeah.
How does it happen, and why is it often delayed in its onset?
PTP is a distinct severe thrombocytopenia, a dangerously low platelet count that typically develops 5 to 12 days after platelet concentrate, or rarely RBCC transfusion. It predominantly affects women. It's characterized by the presence of platelet-specific alloantibodies in the recipient, most frequently anti-HPA-1A in individuals who are HPA-1A negative and have been previously alloimmunized.
So they make antibodies against platelet antigens.
Exactly. The delayed onset is thought to be due to a secondary boosted production of these alloantibodies after the transfusion. The exact mechanisms for the destruction of the patient's own platelets, in addition to the transfused ones, remain somewhat unclear, making it a challenging phenomenon.
How is PTP managed, and are there specific challenges in treatment, especially if additional platelet transfusions might paradoxically not help?
Management is mostly supportive, but may require treatments like IV-AG, steroids, or plasma exchange. Interestingly, and counterintuitively, additional platelet transfusions may actually worsen the thrombocytopenia or show poor platelet increments because the new platelets also become targets. Prevention of recurrence involves using washed blood components, or ideally, components from HPA-compatible donors in the future. 4.2.7, alloimmunization, and platelet refractoriness.
Alloimmunization isn't just about triggering acute hemolytic reactions, is it? It seems to have much broader and longer-term implications for how a patient responds to future transfusions, potentially impacting efficacy.
It's a key and pervasive concept. A recipient can become alloimmunized to numerous antigens on cellular elements and plasma proteins. Allo antibodies to RBC antigens, once detected, can significantly complicate or delay finding compatible products for future transfusions.
Especially for women.
Yes, for women of childbearing age, sensitization to RBC antigens, like DCE, Kell, Duffy, carries a serious risk of causing HDFN in future pregnancies. Antigen matching is currently the only pre-transfusion way to directly prevent RBC alloimmunization, which occurs around 100 times per 100,000 RBCC transfusions.
And what about platelet refractoriness specifically? How does alloimmunization impact platelet transfusions, making them less effective?
Alloimmunization to antigens on leukocytes and platelets, most often anti-HLA antibodies, can profoundly lead to refractoriness to platelet concentrate transfusions. This means a low or absent rise in platelet count after transfusion, despite getting enough platelets.
So the platelets get destroyed quickly.
Right. Once alloimmunization develops, HLA-compatible, cross-matched PCs should be preferred if available. If not, repeated PCs at shorter intervals might be tried, but often less effectively. Importantly, using leukocyte-reduced cellular BCs has significantly reduced the incidence of HLA alloimmunization. Platelet refractoriness can also result, less commonly, from anti-HPA alloimmunization. Other recipient factors contributing include fever, splenomegaly, bleeding, DIC, and certain drugs. Interestingly, cold-stored and cryopreserved PCs have shown preserved hemostatic function even with poor platelet count increases, highlighting that counts aren't the whole story for efficacy. 4.2.8 immunomodulation.
This is a subtle but potentially significant effect. How does the transfusion of allogeneic blood affect the immune system in a broader sense, beyond just specific antibody reactions? It sounds like it could have profound, maybe even unintended long-term impacts.
Transfusion of allogeneic non-self blood can indeed be associated with phenomenon of immunosuppression. This was actually observed early on due to its beneficial effect on kidney graft survival. It seemed to help prevent rejection.
So it dampened the immune system.
That was the observation. The exact intensity and mechanisms are still debated, and it's likely significantly attenuated by using leukocyte-reduced components today, as many effects are linked to donor leukocytes. Transfusion-related immunomodulation, or TRIM, is thought to be primarily mediated by donor leukocytes, either directly transfused or undergoing apoptosis during storage and releasing signals. However, even leukocyte-reduced components can still release immunomodulatory mediators during storage. These mediators, along with the transfused cells themselves, can exert various, sometimes opposing immune effects in vivo, both immunosuppression and inflammation. It's a complex interplay, still an active area of research, with potential implications for outcomes like infection risk or cancer recurrence. 4.3 non-immunologic transfusion adverse reactions. Beyond the immune system. 4.3 non-immunologic transfusion adverse reactions. 4.3.1 fluid overload.
TUCO, transfusion-associated circulatory overload, is increasingly recognized as a major concern, isn't it? As you mentioned earlier, it's now actually a leading cause of transfusion-related fatalities in many places.
Yes, you're absolutely right. Transfusion-associated circulatory overload, TUCO, is common and often under-recognized, yet its clinical impact is profound. Estimated frequencies vary widely, from maybe 10 up to 1,000 per 100,000 components. Critically, and perhaps counterintuitively for many, TUCO is now the main cause of death from transfusion in many developed countries, especially since trial A risk has been mitigated. It's a fundamental shift in the risk landscape.
What patient factors put an individual at higher risk for TUCO? Who is most vulnerable to this type of fluid overload?
Several patient-specific risk factors predispose individuals to TUCO. These include older age, pre-existing renal failure, already having fluid overload, underlying cardiac dysfunction like heart failure, giving large volumes of blood components quickly, and basically an excessive transfusion rate relative to the patient's hemodynamic tolerance.
What are the symptoms?
Symptoms typically include acute dyspnea, shortness of breath, hypoxia, low oxygen, bilateral lung infiltrates on chest x-ray, often alveolar, frequent systolic hypertension, and elevated BMP, that marker of cardiac stress. Fever can also be present, confusing the picture.
How can this dangerous overload be prevented and effectively treated once it occurs? Seems like careful management is key.
Prevention involves carefully identifying at-risk patients beforehand, implementing close monitoring during and after transfusion, ensuring a slow transfusion rate, for example, 1 RBCC over 3 to 4 hours, not rushed, and judicious use of diuretics in stable patients with a history of TACO. If TACO occurs, treatment is immediate. Stop the transfusion, give oxygen, and administer diuretics promptly to remove excess fluid. Severe cases might need ventilator support. 4.3.2 massive transfusion-associated reactions and electrolyte acid-base abnormalities.
When a patient receives what's termed a massive transfusion, defined maybe as more than 5-10 units of red cells plus other components quickly, what specific unique risks emerge from that large volume and rapid administration? Sounds like a major physiological challenge.
It absolutely is. This scenario introduces several unique and critical physiological complications: citrate toxicity, hypothermia, hyperkalemia, and dilutional coagulopathy. These are direct consequences of the volume and nature of the transfused products given rapidly.
Let's break those down. First, citrate toxicity. How does the anticoagulant used in blood bags cause problems in massive transfusions, given it's typically safe in single units?
Citrate, used to anticoagulate blood by binding calcium, becomes problematic when large amounts are infused quickly. The body's ability to metabolize it, mainly in the liver, can be overwhelmed. This leads to hypocalcemia, low calcium, manifesting as tingling around the mouth, circumoral paresthesia, and more seriously, changes in cardiac function, even arrhythmias.
So they might need calcium.
Yes, although citrate is quickly metabolized to bicarbonate, calcium infusion via a separate Bovie line may be necessary, especially with impaired liver function.
And hypothermia seems like an obvious risk, given blood is stored cold in the fridge. How is that managed during massive transfusion?
It is a significant risk. Rapid transfusion of components still at 4 degrees C can cause hypothermia. This drop in core body temperature can lead to cardiac dysrhythmias and also impair coagulation, as clotting doesn't work well when cold. To prevent this, an inline warmer is routinely used to warm the blood components closer to body temperature before infusion. Simple, but effective.
Hyperkalemia also seems like a risk, especially with older units of blood, where cells might leak potassium. How does that develop, and what are the clinical implications?
It is a critical concern, particularly in vulnerable patients like neonates or those with renal failure. During storage, RBCs leak potassium into the storage solution, increasing the potassium concentration over time, especially with longer storage and after irradiation.
Hypocasia. Bad for the heart.
Yes, hyperkalemia can lead to acute cardiac toxicity and arrhythmias. Treatment includes insulin, glucose, calcium gluconate, and furosemide to shift or remove potassium. Prevention involves using washed or plasma-reduced RDCs, using fresher units, for example 7-10 days old, and avoiding units stored 24 hours after irradiation. 4.3.3 iron overload.
With patients who receive frequent long-term transfusions, maybe those with chronic anemias like thalassemia, iron overload must be a major cumulative concern, right? Since the body has no easy way to get rid of excess iron.
Indeed, this is a pervasive and serious long-term complication. Each unit of red blood cells contains a significant amount of iron, specifically 200-250 milligrams. In frequently transfused recipients, this iron accumulates progressively. If left untreated, it affects multiple vital organs: endocrine glands, the liver, causing dysfunction, cirrhosis, and critically, the heart. Ultimately, patients can die from cardiac failure or arrhythmias due to iron deposition in the heart muscle.
How is iron overload monitored and effectively managed to prevent these devastating long-term consequences?
Iron overload is meticulously assessed using serum ferritin measurements, an indicator of stores, and more directly by MRI of organs like the liver and heart to quantify iron deposition non-invasively. Liver biopsy can also be used. Prevention and treatment rely on careful, ongoing monitoring and timely initiation of iron chelation therapy, using drugs that bind iron so it can be excreted. It's often a lifelong commitment for these patients. 4.3.4 hypotensive reactions.
Acute drops in blood pressure seem like a simple, non-specific reaction that could happen for many reasons during a transfusion. but the source flags a specific type of hypotensive reaction that has a particular interesting cause.
That's right, there's a distinct pattern. Acute hypotensive transfusion reactions are defined as an abrupt drop in systolic blood pressure, maybe 30 millimilli Hg, occurring early after the transfusion begins, typically within minutes. What's characteristic is that they resolve quickly once the transfusion is stopped, usually without needing further medical intervention. Other mild symptoms might be present too.
How often does this happen?
Estimated frequency is low, 110 per 100,000 components. These reactions are thought to result from the generation of vasoactive kinins, like bradykinin, in the blood components during storage or contact activation.
And certain patients are more prone?
Yes, they are significantly more likely in hypertensive patients taking angiotensin-converting enzyme ACE inhibitors. These drugs impair the breakdown of bradykinin, allowing it to accumulate. If this specific reaction occurs, the same blood product shouldn't be restarted. Switching from an ACE inhibitor might be considered for future transfusions. 4.3.5 adverse transfusion reactions of uncertain imputability.
And finally, the text mentions some reactions are still being investigated for a clear, definitive causal link to transfusion, meaning the evidence isn't fully conclusive yet. This highlights the ongoing complexity of research in this field.
Yes, exactly. Two examples are mentioned that illustrate this challenge. First is necrotizing enterocolitis, a severe intestinal condition common in preterm neonates. NEC has been infrequently described with a close temporal association with RBC transfusion. However, direct causality remains to be definitively ascertained. It's not proven transfusion causes it. The efficacy of withholding feeds during transfusion to prevent it is also debated. Second is posterior reversible encephalopathy syndrome, PRES, a rare neurological syndrome with acute reversible symptoms due to brain edema. It's been described within 10 days after RBCC transfusion, mainly in women with severe long-standing anemia. Prognosis is often favorable, but irreversible damage can occur. Prevention might involve avoiding overly rapid correction of chronic anemia. Again, direct causality isn't firmly established. 4.4 infectious adverse reactions, the ongoing battle against transfusion-transmitted infections.
The fear of getting an infection from a transfusion is a big concern for many people, especially historically. How is that risk managed today, and how rare are these events now with modern screening?
This is an area where transfusion medicine has made truly monumental strides. Donor screening is the first and arguably most critical line of defense. Rigorous selection based on questionnaires, lifestyle, medical conditions, exposure risks.
Okay.
Then, once blood is donated, it undergoes extensive multi-layered testing for infectious agents, either detecting antibodies, serology, or directly detecting the infectious agent's genetic material, most often via highly sensitive nucleic acid amplification testing.
NAT testing.
That's very sensitive, extremely sensitive. The increasing sensitivity of these methods has progressively narrowed the window period, that early phase after infection when a virus might be present but undetectable by older tests. This significantly reduces the residual risk to astonishingly low levels for many viruses.
What about bacterial infections specifically? Are they still a concern given all the viral screening? And how do they manage the risk of bacteria getting into what should be a sterile product?
Transfusion-transmitted bacterial infection remains a significant concern, especially with platelet concentrates because they're stored at room temperature, 20-24 degrees C. This allows bacteria to proliferate if contamination occurs during collection or processing.
So room temp storage is the issue for platelets.
Primarily, yes. Red cells, stored at 4 degrees C, can also be implicated, but less often as growth is slower. Recipients of contaminated components can develop abrupt fever and chills, potentially deteriorating rapidly to septic shock, DIC, and death. Endotoxins released by bacteria can play a major role. Prompt initiation of broad-spectrum antibiotics after taking cultures is critical if contamination is suspected.
Is there a way to neutralize pathogens right in the blood product itself as a kind of final fail-safe before it even gets to the patient?
Yes, that's where pathogen reduction technologies come in for platelets and plasma and potentially for red cells in the near future. These offer an additional layer of safety. They use various methods like chemicals or UV light to inactivate a wide range of viruses, bacteria, and parasites within the component.
Are they completely effective?
Highly effective for many pathogens, but it's important to note they're often ineffective against bacterial spores and certain non-enveloped viruses like hepatitis A, parvovirus B19, and hepatitis E. Another safety measure is post-donation information. If a donor reports illness shortly after donation, the products are quarantined.
Given all these layers, screening, testing, pathogen reduction, how incredibly rare are transfusion-transmitted infections now? And what about emerging unknown threats? How does the system adapt?
They are indeed increasingly rare, a huge success story. However, the field must remain constantly vigilant because the emergence of new or previously unidentified infectious risks is a persistent threat. We saw this with West Nile virus and babesiosis in the US and hepatitis E in Europe.
So new threats pop up.
They do. This necessitates active surveillance programs and swift implementation of mitigation measures, maybe new tests, broader pathogen reduction, new donor deferral criteria based on travel. Other arboviruses like dengue, Zika, chikungunya are also monitored. Documented transfusion cases are rare, often lacking clinical impact, but the potential exists. It's complex. Whether infection occurs depends on pathogen dose, virulence, survival in the component, storage conditions, and the recipient's immune status.
Table 11326 provides a comprehensive look at the estimated frequencies for these TTIs, really putting the current risks into perspective. Could you highlight some key examples that illustrate just how effective current measures are?
Absolutely. The table really shows the impact of safety measures. For pyogenic bacteria in platelets, donation prevalence might be 10-20 per 10,000 before mitigation. But with bacterial detection and/or pathogen reduction, recipient sepsis drops from maybe 530 per million PCs to less than 0.5 per million. Huge reduction. For HIV-12, donation prevalence is tiny, maybe 0.1 per 10,000. Serology plus NAT pushes the residual recipient risk down to an incredible 0.1 to 1 per million units. Similarly, for hepatitis B and C, NAT has dramatically cut recipient risk to below 0.5 per million for HBV and maybe 0.11 per million for HCV. CMV is common, but leukoreduction reduces recipient infection risk to less than 0.1 per million, vital for vulnerable patients. West Nile virus in endemic seasons might be up to 3 per 10,000 donations, but NAT reduces recipient risk to less than 1 per million there. Malaria, plasmodium varies hugely by region, but screening and deferrals keep recipient risk below 0.1 per million in non-endemic areas. The variability highlights how local factors, rules, and reporting influence these numbers, but the overall picture is one of extremely high safety due to continuous effort.
The horizon alternatives and future directions in transfusion medicine.
Looking ahead now, it seems the field is moving beyond just ensuring safe transfusions to also proactively reducing the need for them wherever possible. What are some of these transfusion-sparing strategies that are being actively adopted in modern medicine?
Patient blood management (PBM) programs are absolutely key here, driving a paradigm shift away from reflexive transfusing towards optimizing the patient's own resources first. They advocate for several crucial transfusion-sparing strategies. First, aggressively treating anemia and/or iron deficiency before any planned surgical interventions.
Get the patient ready beforehand.
Exactly. Optimize their own red cell mass and physiological reserve. Second, meticulously minimizing blood loss during surgical interventions through advanced techniques, careful hemostasis, and drugs that reduce bleeding. And third, optimizing the patient's intrinsic tolerance to anemia, allowing clinicians to manage stable patients safely at lower hemoglobin levels, avoiding unnecessary transfusions based just on a number.
Are there pharmaceutical alternatives that can directly stimulate the body's own blood production, effectively reducing or even eliminating the reliance on donor blood for some conditions?
Yes, absolutely. Erythropoietin, EPO, is a prime example. It's a hormone that stimulates red blood cell production, and synthetic versions are widely used in patients with anemia from chronic renal failure and other conditions, reducing or avoiding transfusion needs. Similarly, thrombopoietin, TPO receptor agonists, have shown significant promise in stimulating platelet production, reducing platelet transfusion needs, particularly after chemotherapy.
This is truly exciting. What about cutting-edge therapies that could really transform transfusion medicine, potentially moving us beyond conventional blood products entirely, or even, creating blood outside the body?
The horizon is indeed very promising. Gene therapy approaches in patients with chronic hemoglobinopathies like sickle cell disease or major thalassemia offer the profound potential to dramatically reduce or even eliminate their lifelong transfusion needs by correcting the underlying genetic defect.
Curing the disease itself.
Potentially, yes. And even more futuristically, stem cell-derived blood cells like red blood cells or platelets grown in a lab from pluripotent stem cells may one day become a viable and perhaps universal alternative to relying on volunteer donors, especially for rare blood types or ensuring a continuous supply.
So the focus isn't just safety anymore, but also deeply understanding the measurable efficacy of transfusions and finding completely new ways to achieve the same goals, maybe even manufacturing blood components.
Precisely. The issues surrounding transfusion safety, while always paramount, have evolved to now fully encompass transfusion efficacy. There's a recognized and pressing need for new means of assessing transfusion efficacy beyond just simple cell counts or hemoglobin levels. We need to understand the true clinical benefit, long-term outcomes, quality of life.
And how will we understand this efficacy better, perhaps on a large population-wide scale? What kind of data and research will be needed to truly define optimal transfusion care in the future?
Large-scale biological and population-based databases pertaining to both blood donors and transfused patients will be instrumental. These comprehensive data sets will allow for assessing and understanding the fundamental basis of transfusion efficacy, enabling large-scale, real-world data analysis and identifying subtle impacts or variations in response. This raises an important question for the future.
Yes.
What new, perhaps highly individualized criteria will truly define optimal transfusion care in the years to come? Optimal care may soon require considering criteria related not just to the recipient's immediate needs, but also to a deeper understanding of the donor's specific genetic and immunological profile and the precise characteristics of the blood product itself.
Yeah.
It reflects the incredible complexity and evolving nature of this field, moving towards an era of highly personalized transfusion medicine. Ouch.
Wow. We've truly taken a deep dive today into the fascinating and clearly incredibly complex world of transfusion therapy and biology. From the meticulous processing of blood components and that intricate dance of antigens and antibodies, to the critical safety measures protecting every patient, and those exciting future innovations, it's absolutely clear this field is a cornerstone of modern medicine.
We've seen how a seemingly simple act like giving blood involves profound biological understanding, rigorous quality control, constant vigilance against a myriad of potential adverse reactions, immune, non-immune, and that persistent evolving threat of infections. The journey of a single unit of blood is really a marvel of science and logistics.
And we've peeked into the future where patient blood management, groundbreaking gene therapies, and maybe even stem cell-derived blood cells promise to fundamentally reshape how we think about and deliver transfusion care. It's a field that's clearly always pushing the boundaries, saving and improving countless lives.
The key takeaway here, I think, is the dynamic nature of knowledge in this field. As we understand more about the human body's intricate interactions with transfused components, our ability to provide safer, more effective treatments just continues to grow exponentially. This deep dive should arm you with an understanding that goes far beyond the basics, giving you profound insight into the sophisticated strategies ensuring transfusions save lives daily.
So what does this all mean for you, our listener? I hope you now feel not just well-informed, but truly empowered with a deep, almost professional level understanding of what goes into every single drop of transfused blood. Next time you hear about a blood drive or a patient needing a transfusion, you'll know the incredible science, the meticulous process, and the unwavering dedication behind it. You'll understand it like the elite professionals in the field aim to.
Indeed. And consider this final thought. As our understanding of individual genetic profiles expands, how might precision transfusion medicine become the norm? Tailoring every blood component not just for ABO and RH, but for a much wider array of factors based on the patient's unique genetic and immunological blueprint, to an even greater degree than we achieve today, making transfusions even safer, even more effective. It's an exciting future.